Welcome and hi everyone. Today we are exploring chromatography through a classic and colorful application, the separation of plant pigments. By definition, chromatography is an analytical technique that separates the component of a mixture based on how they interact with two different phases. In our plant pigment experiment, these two phases are, first, the stationary phase, the fixed phase, which is typically the chromatography paper or a TLC plate. The second one is the mobile phase, the solvent or liquid developer that move up through the stationary phase. Our mechanism of separation relies on the fact that different plant pigments have unique chemical structure and polarities. Because of this, they travel up the paper at different rates based on their affinity for the solvent versus the paper. While chromatography has many common variants, including paper, thin layer, gas, and liquid chromatography, the significance of this technique is massive. Its versatility, accuracy, and efficiency make it a foundation method in modern laboratories, allowing us to identify, purify, and analyze complex biological mixtures just like the pigment that power photosynthesis.
Assalamualaikum, my name is Nur Alia Maisara binti Abdul Rahman. Now I'm presenting about theory of chromatography. Chromatography separates compounds according to differences in polarity and solubility. In paper chromatography, the paper acts as stationary phase, while the solvent acts as the mobile phase. When the solvent moves up the paper by capillary action, the pigments travel at different rates. Non-polar pigments are more soluble in the solvent and move further, while polar pigments interact more strongly with the paper and move a shorter distance. The retention factor RF value can be calculated using formula RF equal to distance traveled by pigment divide distance traveled by solvent form. Different pigments have characteristic RF value that help in their identification.
Now, I will explain the methodology used in this experiment. 1. Fresh green leaves are crushed using a mortar and pestle. 2. A small amount of solvent ethanol is added to extract the pigments. 3. A spot of the extract is placed near the bottom of chromatography paper. 4. The paper is suspended in a chromatography chamber containing a suitable solvent mixture. 5. The solvent rises through the paper and separates the pigments. 6. After separation is complete, the paper is removed and allowed to dry. 7. The distance traveled by each pigment and the solvent front is measured to calculate RF value. This slide shows the example of the result obtained from paper chromatography. 4 pigments were successfully separated from the leaf extract: carotene, xanthophyll, chlorophyll A, and chlorophyll B. Carotene appears orange and has the highest RF value of 0.95, meaning it's traveled the furthest because it is the most soluble in the mobile phase. Xanthophyll is yellow with an RF value of 0.71, followed by chlorophyll A is blue-green with an RF value of 0.65. Chlorophyll B is yellow-green with the lowest RF value of 0.45, so it's traveled the shortest distance. Paper chromatography separates pigments according to their solubility and attraction to the stationary phase. RF is calculated by dividing pigment distance by solvent front. Now, I will explain the difference between the mobile phase and the stationary phase in paper chromatography. The mobile phase is a phase that moves over or through the stationary phase, carrying the analyte mixture. It is also called as the eluting fluid. Its function is act as the carrier for the mixture or sample to pass through the stationary phase, so it is known as the moving part. In this experiment, the mobile phase is a liquid solvent. In contrast, the stationary phase is a phase that is fixed in place in a column or on a planar surface. It is a porous solid used alone or coated with a stationary liquid phase. Its function is to absorb the component being separated, so it is known as the static part. In this experiment, the stationary phase is in the form of gas or liquid that move over the stationary phase. because different pigments interact differently with the paper. They move at different speeds, producing separate colored bands. This difference allows
Next, I will talk about adsorption versus partition of chromatography. In adsorption chromatography, separation is based on surface attraction, which is it happens because compounds stick to the surface of the stationary phase. For the movement, if the attraction is strong, the compound will move slower. The example of adsorption is thin layer chromatography. In partition chromatography, separation depends on how soluble the compound is between two phases. For the movement, if the compound is more soluble in the solvent, it will move faster. The example of partition is paper chromatography. Overall, adsorption is about surface attraction, while partition is about solubility. Paper chromatography versus layer chromatography. Paper chromatography (PC) and thin layer chromatography (TLC) are both used to separate compounds, but they have some differences. Paper chromatography uses paper as the stationary phase and works based on partition. It is slower and cheap, but gives lower resolution. The sensitivity is less than thin layer chromatography. Also, it was used in sample analysis like plant pigments. Thin layer chromatography uses silica or alumina and works based on adsorption. It is faster and more sensitive and gives better separation, but it is slightly more expensive. It is also more accurate in lab analysis. In short, PC is simpler, while TLC is more efficient and accurate.
Now, let's look at the two different ways we can set it up physically, column and planar. In column chromatography, the stationary phase is packed inside a glass or plastic tube. The mobile phase flows through the column, usually driven by gravity or pressure. This is a powerful method for purification and isolation. For example, if we wanted to isolate a large amount of pure chlorophyll A from a leaf extract to use in a separate experiment, we would use a column chromatography. On the other hand, for our analysis of plant pigments, we are using planar chromatography. In this method, the stationary phase is spread on a flat surface, such as paper or a glass plate. Instead of flowing through a tube, the mobile phase moves across the surface via capillary action. Planar chromatography is the standard choice for identification and purity analysis. It's what allows us to see those beautiful distinct bands of green, yellow, and orange on our paper strip, making it easy to identify each pigment present in the plant. Next, let's look at the chemical behavior of our chromatography system. We'll classify this into normal phase and reverse phase, which is entirely decided by polarity. Essentially, which phase is hydrophilic and which is hydrophobic. In normal phase chromatography, the stationary phase is polar, like standard filter paper or silica gel, while the mobile phase is non-polar, because polar compounds get held back by the stationary phase, while non-polar compounds race ahead with the solvent. When separating plant pigments in a normal phase system, the highly non-polar pigments like yellow, orange carotenes will travel the fastest and furthest up the paper. Meanwhile, more polar pigments like chlorophyll B will bind tightly to the paper and stay closer to the bottom. Reverse phase chromatography is the exact opposite. Here, the stationary phase is non-polar and the mobile phase is polar. This completely flips the illusion order. Polar compounds move the fastest while non-polar compounds are retained longer. Understanding this distinction is key because it tells us exactly how to read our result and identify which pigments is which based on how far they travel.
The conclusion for this presentation is chromatography analysis is an effective technique for separating and identifying plant pigments. The method works by exploiting difference in polarity and solubility among pigments. Through chromatography, a pigment such as chlorophyll A, chlorophyll B, and xanthophyll and carotene can be clearly separated and analyzed. These techniques provide valuable information about plant composition and has many applications in scientific research and chemical analysis.